I actually don't think I'll be able to see much with these on. So, G'day scientists! Today we're reviewing two DNA extraction kits. This one here from Thames and Kosmos and this one here from Science Lab. I am super excited because I have never ever extracted DNA from anything in my life. We are definitely going to learn something today. First up, we're reviewing the Genetics and DNA Lab from Thames and Kosmos. It cost me $45.41 from Amazon. Right, let's open up and take a look. First of all, a massive danger sign on the top. It's saying denatured alcohol, also known as blah blah blah, is highly flammable. Does this have alcohol in it already? First up, this looks like protein strands to go on DNA here. I guess this is to build the DNA model itself. Then we have some Petri dishes, denatured alcohol. So the denatured alcohol isn't included because the lid's not on it. That's a bit of a surprise. Let's hope it wasn't in there and it's all leaked out in there in the shop. Probably not. We probably have to fill that up with denatured alcohol. There's a book here which we'll look at in a tick. I'm guessing this is to hold test tubes, a little test tube stand. A poster with a cross section of an animal cell. Some DNA. Looks like this is like a forensic analysis sheet. That is it in the box. First up, I have to think there's not a lot in the box. This kit here. This one here has got LB agar, which is the stuff you put onto a Petri dish to grow specimens. So let's open up the book and have a look. It's actually a really thick book. Learning how to solve a crime, taking fingerprints, genetic engineering. Let's get to the main experiment, which is extracting the DNA. So this is it here. Find the DNA in a tomato. Wow, it's a several step process. First you will need an empty brown bottle, this cap here. Then we need a plastic spatula, denatured alcohol, methylated spirits. Now I happened to go to our local hardware store this morning to buy some methylated spirits, otherwise I wouldn't have had it in my house. It's quite a big ask and probably quite a dangerous substance to use with a young scientist, so this is definitely one for supervision. Here is the denatured alcohol or methylated spirits. You can see it's got this giant poison sign down the side. Right, there's the alcohol. Next we need some table salt, dish soap, spoon, knife, tomato, a freezer, a blender, and two large empty yogurt containers. One yogurt container should be large enough for the hand blender to fit inside of it. Okay, I've raided the kitchen and fetched all the things we need for the first part of this 4-stage experiment to extract DNA from a tomato. First step, the night before your experiment. For real, and they're just telling me this now as I'm sitting down to do the experiment. It says fill the brown bottle with the denatured alcohol and place it in the freezer to get it nice and cold before the experiment. Now methylated spirits is flammable, poisonous, and not good to breathe in, so you must ensure that you're doing this in a well ventilated space. I have the window open here, which is why you can hear the crickets in the background. Okay, it's full up and overflowing. So I'm going to clean this toxic mess up and come right back to you. It's the next day. The alcohol's been in the freezer overnight and I'm ready to get started with the first phase of the experiment. Now the first phase of the experiment says to cut a tomato in half and then cut that half into small pieces. Now the title of this video is how to extract genetic material from fruit. So you might be wondering why I'm cutting a tomato. If you know the answer, put it in the comments below. Now we've chopped the tomato, you place it into the tub. In the other container, we need to put 50 milliliters of dish soap, which is twice this pink measuring cup, and 1 teaspoon of the table salt. Stir everything well with the spatula and pour it over the finely diced tomatoes. So here's the spatula. Gonna give it a stir. Whoa, I'm already splashing stuff everywhere. You'll definitely need to have a cloth with you when you perform this experiment, and it's a shame that the book isn't waterproof because I've already spilt tomatoes and washing up liquid and salt all over it. Next step is to pour the dishwasher soap with the salt mixed into it over the top of the tomatoes. Now we take the stick blender and we blend the tomato, sphaery liquid and salt together. Now there was a warning of this part of the experiment. It says that it's going to get pretty frothy and will likely spill all over the place. I'll try my hardest. This is a really high speed hand blender, so it will be interesting to see what happens. Here we go. Now I can see almost all of the tomato pieces have been completely blended now. This mixture is incredibly frothy. Okay, now we're on to step 2 of the experiment, and for this part of the experiment we need the tomato washing up liquid salt blended combination we made earlier, a test tube, a funnel, some filter paper, a permanent marker pen, the ruler, and this piece of cardboard which we're going to fold into a test tube stand. Now what's annoying with this test tube stand is that it's got all these tabs for folding together, but there's no glue included in the kit, so I'm just going to use some sellotape to hold it together. Normally there's like little slots that you put the flaps into, but they don't seem to be on this. Now you can see I've just added 3 pieces of tape to this test tube stand to hold it together. Then we place the funnel into the top of the test tube. Now we fold the filter paper in half, create a little funnel, and this goes into the top of the funnel. Now we're going to take the ruler, and with permanent marker pen we're going to measure 3 centimeters from the bottom of the test tube. Then mark another line 3 centimeters above. It will take a while for the clear liquid to fall drop by drop into the test tube. Keep adding the mush until the liquid in the test tube reaches the first mark. So let's add some of this in. So far, no drops. I think this might take some time. We're going to leave it to drip and I'll come back in 10 minutes. Hey, it's been 30 minutes and I've come back to find that indeed some of the drops have started falling through the filter paper and into the bottom of the test tube. I have also noticed in the tub that we were storing the mush that this separation has started to occur naturally. What I'm going to do to speed the process up is filter off some of this foam on the top and put it onto the plate so that we can access this settled solution at the bottom and add it directly to the top of the test tube. Here we go. Now you can see most of this tub is filled up with the solution. I'm going to pour that into the top of the filter paper. I've come back 30 minutes later and I can see already that more than we need has been filtered into the test tube, which is great news. I'm going to pour a little bit out to get it up to the 3 centimeter mark. This is the most exciting part of the experiment because we're actually going to make the DNA become visible. Okay, for this part of the experiment we need the denatured alcohol that we placed into the freezer yesterday, the test tube with the 3 centimeters of tomato mush filtered, and a pipette. For this stage of the experiment we're taking alcohol into the pipette and dripping it slowly down the side of the test tube until we get to the 3 centimeter mark that we added above it. And I don't know if you can see already, we can, yeah, if you have a look very closely at this, we can start to see the DNA forming and being extracted on the top there. Now we're going to the next part of the experiment, which is taking the stopper and very slowly inverting the mixture and then putting it back again. As we do this, we can see more and more and more of the DNA being extracted and being taken to the surface. Now we're going to take the wooden stick and pull the DNA out of the solution. If you can see that on the camera there on the end of the stick, dyed red by the remainder of the tomato in the liquid, that's the DNA stuck to the stick. Now this kit, it was quite expensive for the parts contained within it. The experiment itself to extract DNA was quite slow and involved quite careful measurements with hazardous chemicals. It turned out okay, we got DNA out of it. But was it worth the money? We had to provide a lot of the ingredients ourselves, including using a hand blender, which is quite full on for an experiment for a young person. The kit does contain other experiments, but does that warrant the higher price for what is quite an empty kit? I'm going to give the Genetics and DNA Lab from Thames and Kosmos 2 stars for fun, 1 star for price, and 2 stars for learning. Buy or bye-bye? It's a bye-bye. Just not enough value in this kit to make it worth the money. Okay, next up we have the DNA extraction set from Science Lab. Let's open up the box and see what's inside. First, right away, some safety goggles. These would have been super useful for the last experiment where I spilled denatured alcohol everywhere. Let's open them up and see if they fit an adult as well as a kid. Normally these things only fit children and they're a little bit tight. I actually don't think I'll be able to see much with these on, so definitely not adult size, but we'll probably be okay for a young scientist. We will proceed without the goggles. Next up we've got some test tubes and filter papers. Next up we've got the instruction leaflet. It's small and a little bit creased, but it's made from plastic coated paper, which is good because last time we were spilling things everywhere and stuff was getting a bit messy. Then we've got some stirrers and some tweezers and a pipette. We have a test tube stand made from plastic, a funnel, a magnifying glass. Now these magnifying glasses are usually pretty useless. I have some gloves. Again, these would have been useful in the previous experiment where I spilt things everywhere. Let's try them on, see if they fit an adult. And I've got quite small hands though. They do fit an adult. Some plastic bags, a measuring cup and some test tubes. Okay, pour 40 milliliters of alcohol into the small beaker and store it in the freezer. The alcohol will need to be freezing cold when we perform the experiment. It's exactly the same as the last one. So if you're doing these with young scientists, make sure that you sneak in, put some alcohol in the freezer before you go to start the experiment, because having to wait 24 hours to start would be quite annoying. This is the beaker that they want us to put it into. There's no lid, so we're going to put 40 milliliters in, which is up to the measuring line, and then have to just keep it in the freezer uncovered. I am going to get some cling wrap to put around the top to make it a bit safer. Now you can see this has warning signs all over it. It's very dangerous, it's flammable, you need to use it in a well ventilated space and store it appropriately, and definitely supervise young scientists. We're going to come back in 24 hours time when this is frozen and move on with the rest of the experiment. Okay, for the first stage of this experiment, we need the plastic beaker that came in the kit, some washing up liquid, some salt, and the measuring stick. The first step is to add 10 milliliters of washing liquid to this cup. There is no 10 millimeter mark on this cup, so I'm going to add it to roughly 10 millimeters. Now we have 10 milliliters in this cup. We have to add 1 milliliter of salt. Now we use this spoon to dissolve the salt into the washing up liquid. Now, as before, you're going to need a cloth because this gets pretty messy. Adding the salt to the washing liquid makes it more watery. So while stirring, I've splashed liquid onto the instructions, which luckily are wipe free. For the next step, we need the liquid we prepared earlier, washing up liquid and salt, which is a lysis buffer, the strawberry, which we're going to cut into small pieces, and a plastic bag. Now we've chopped up the strawberry a little bit to get started. We add it to the plastic bag. Now we're adding 20 milliliters of the lysis buffer, which is actually all of it because we only added 10 milliliters of soap to begin with. So let's pour all of it in. Now we squeeze as much air out of the bag as possible and seal the zipper shut. Then with our hands, squish the bag up. What we're trying to do is, like the blender in the previous experiment, just mix the strawberry into the buffer. And we have to do this for a few minutes. So while I'm mushing the strawberries up, if you found this video useful, please smash the like button. If you want to know which science kits to buy, please subscribe to the channel and hit the notify button so you can be notified when I next drop a video. It's now been 2 minutes and I've mushed the strawberries up into this mushy consistency. There's a few tiny bits remaining, but enough of the strawberry mixed into the lysis buffer. For this next step, we need the mushed up strawberry, a test tube, the pieces of the test tube rack, a funnel and a filter paper. Then we put the funnel into the top and fold the filter paper into 4, add the filter paper into the funnel, then pour in the mushed up strawberry from earlier. The great thing about this experiment is it takes the entire amount of mushed up strawberry we put in and there's less foam, so we don't need to filter it off the top. I'm hoping this means that the strawberry liquid will drop through the filter paper faster. I'll come back in 10 minutes. Oh my goodness, I just reread the instructions and realised I've completely messed this up. I was supposed to have added 100 milliliters of distilled water into the lysis buffer before I added the strawberry and mushed it up. And that's why nothing is dripping through. So I'm going to add the strawberry liquid back into the bag. I'm going to add 100 milliliters of distilled water into the bag, add the filter back into the top of the test tube and pour some of the liquid in. Now I expect what's going to happen is this is going to filter through much faster than it was before. I'm going to come back in 10 minutes and see what's happened. Okay, it's been about an hour now and the liquid is almost finished dropping through into the test tube, so I'm going to take the funnel out. The next step of the experiment is to take the alcohol that we placed in the freezer the night before. We take 5 milliliters of alcohol and we're dripping it down the side of the test tube very slowly so that it doesn't mix but forms a layer. There we go. You can see very, very, very delicate, fine strands floating in the liquid, and that's the DNA. Now this was the cheaper of the two kits, and actually was better value for money as a result. The parts in this kit are mostly plastic, and to be honest, you could find most of these around the house. So that combined with the other things you needed to combine, you begin to question the value of this kit. I learned quite a bit from doing it, and it was actually quite good fun, and the experiment was simpler than the other kit. I'm going to give the DNA extraction set from Lab Science 2 stars for fun, 1 star for price, and 2 stars for learning. Buy or bye-bye? It's also a bye-bye. You can perform this experiment in your home without buying any of these kits. If you did have to buy one, I would probably suggest this kit over the Thames and Kosmos. That was a bit disappointing. I was super looking forward to extracting DNA, yet none of the kits quite measured up. If you'd like to see a good review, check out this review here where I make the spin art kit from KiwiCo.